Is there any kind of a pointer? Oh, oh this. Okay, good. All right, well, let's see if I can. Okay, so we're going along the same line as the previous speaker in attempting to get simplicity, but we're also trying to use an engineering principle of practicality. And so one of the things we're starting with, I'll tease you, but I really do think what you're doing is extremely important. And one of the suggestions people have made is, well, let's get rid of one codon. You've at least accomplished that, and that's a big job in E. coli. Anyway, I want to start right out and point out the collaborators who've worked on this. There's a group in Szeged, Hungary, Georgi Pastai and his student Farah Tamash. Gunter Keil is from Laboratory of Animal Virology in Germany, which I will get to some very interesting experiments that he happened on that I'm going to speak. And then Sarah Harcum is a fermentation specialist at Clemson University who's been working with us. And then my group in Madison includes all of these people who work for a new company we just formed called Scarum Genomics. This technology was developed in my lab at the university, and it's sort of being technology transferred by these people joining the company. Anyway, another thing my university calls for is that I have to tell you that I have a financial interest in 3 companies in every talk. And now we get to the first slide. E. coli we sequenced with great effort in the very early days, and the genome has just been updated, and I wanted to let you all know that if you go to this website, you'll be able to get a copy of the sequence that has had 375 mistakes corrected. I'm kind of impressed. This was Dr. Horiuchi in Japan who finished totally sequencing the W3110 strain and found some differences, and then went back and compared it with the MG1655 strain that we had sequenced, and he was right on 374 out of 375 differences. But I'm kind of proud of the fact that with the technology that was available at that time, we got to the level of 1 mistake every 10,000 bases. Anyway, the corrected version is up. There's improvement in simplification. There are now fewer open reading frames than there were. The reannotation has merged up some fragments of open reading frame. And there's still 18% of this genome has genes that are of essentially unknown function. Many of them have matches to other genes of unknown function in other organisms, but when we started it was 37% of unknown function, but there's still an awful lot to learn about E. coli. So, one of the things I thought I would do is get to a little bit about what we know about the functions of the E. coli genes. You can classify the ones that are known, and it has basically all the things needed to make a robust life form that can grow on mineral salts medium. And it is also metabolically very general in that it has 2 whole ways of doing things, anaerobic or aerobic. So it can switch back and forth. If it spends most of its time in the guts of animals, which I'm not sure if that's true, what it does is lives right on the outer surface of the inner surface, depending on your point of view, on the surface of the gut wall. And I tend to have an inside view for some reason, speaking of philosophy. Sorry. Anyway, this is serious business. On that inner surface, there's oxygen that is coming in from the capillaries, and the anaerobic bacteria that are in the gut are either shielded by having that oxygen scavenged out, or E. coli is using this as an opportunistic way of gaining an energy advantage over the anaerobes. But actually the regulation is very fascinating right at that margin where it really grows, where both functions can happen at once. And the cell has stuff that even shields some of these enzymes that are damaged by oxygen, things that look like cytochromes that go around them. so that it can do this process of taking advantage of the TCA cycle at the same time as doing anaerobic fermentation. Another thing that E. coli has, and I wouldn't be surprised if the other organism, mycoplasma, has, is chaperones. There's a huge investment made in folding proteins correctly, or at least folding them, and it's not clear whether you could ever get rid of all of the chaperones. And so there's just a contrast. I'm not sure whether it's between the organisms or just between the perception, but there's a huge amount of integration of functions that there's a lot of genes that have 2 or 3 modules of a chemical reaction, possibly so that the substrates don't have to diffuse very far. And I think it'd be very hard to believe that we could understand that except by experiment. And so one of the things I like about this project, which is reducing the genome, is exactly the point that was made before, that we learn a lot of biology as we go along. So I think with a list like this, we decided to pick some things that we really could delete because they really looked unimportant, and then work our way down. One thing that was really interesting to me, and this is a little bit hard slide to explain because it's kind of a statistical nightmare, but basically this is the percentage of the genes for that function that are highly expressed. And it turns out that the genes that are poorly expressed in lab cultures, at least, are the ones we don't know anything about. Duh. A lot depends on what you actually look at. And just starting these anaerobic experiments, we got this huge passel of very highly expressed genes that have a Y in the name, which means they're unknown. So I think it's important to go for a lot of different conditions. And in order to standardize, a standardizable medium is really important. Something like LB gives you a different gene expression pattern every time because every batch of LB has got a different mixture of God knows what. So I think it's not just so that you can make it convenient, but also it's very hard to do the research on gene expression unless you have a very defined way to culture the organism. Anyway, in the course of the research on now sequencing 4 strains of E. coli, we've come up with the realization that there's an ancestral backbone sequence in the genome we call a core that is common basically to all the strains, and you can see it in some rather distant Enterobacteriaceae, fairly rearranged, but the genes are there. The codon usage tends toward a evolutionarily defined consistency in the part of the genome that evolved by descent. That's what the core genome looks like, and what it includes are the essential housekeeping genes, pathways, characteristics of the genus, DNA replication, all the stuff that is really kind of going to contribute to a generic bacteria would probably be in the core genome. And then these islands, which are relatively recently introduced, acquired through horizontal gene transfer, weird codon usages reflecting different origins, and then pathogenic strains contain virulence and pathogenicity genes. Combinations are probably needed to get a pathogen. The sorts of estimates are is that you need to put together about 50 things at random in order to get a pathogen, just judging from the numbers of factors that people begin to study when they actually start taking apart the pathogenicity, the pathogen function. I believe that what these islands really do is confer the ability to grow in a niche. The niche may be living off of some pathogenic process, but also beta-galactosidase and lactoperon is on an island. And the only thing I can think of is that if the genome could get as big as you wanted to, you could probably have every niche, but that isn't the way it works. Whether there's a lack of economy in having a huge chromosome or whatever it is, this thing keeps changing to be able to grow in different niches. Now I'm going backwards there. Then there is a phenomenon which I'm not going to talk about where you have specific hypervariable regions that are intended to foil the immune system, and those, of course, are not conserved in the same sense. When we sequenced 0157H7 and we found these introgressed regions or islands, it was then pretty interesting to see that E. coli K12, the lab strain, also had islands. Those are the red things. And so we came up with this Venn diagram in which the core would be this blue stuff, and then there'd be lineage-specific segments. And the lineage-specific segments in 0157 was really quite huge, 1.2 million base pairs, 20% of the genome. It's a little bit less for K12. Then we did the third genome, which was a uropathogenic strain, and that gives you a three-way Venn diagram. And once you get three of them, you can start seeing that the core begins to be defined now as that which is in common between three. No question there's a lot of things that occur in only one genome, not too much that occurs in just two. So it really looks like each genome has a core and then other stuff that adapts it to a niche. At least that's the way I'm going for. So in this sequencing project, we did 7 million base pairs, and 52% of that total pathosphere is in the core. And as you get more, it gets bigger. And this turned out to be a really useful tool for designing the reduced genome. This is the 4 sequence genomes. I didn't mention what the fourth one is. It's a meningitis strain. And the colored stuff are things that are in that genome that are not in K12. No, things that are, yeah, the baseline is K12. So anything that is homologous to K12, you don't see anything. If it's different, if it's an insertion, you have it. Why don't I get this? I'll hold it. So if you look at these regions and delete anything that is different in all the strains or absent in all the strains, then you have something that can be removed and you're moving toward the basic core. You got to, oh, you want to help?
Yeah.
I need it. So this is kind of the evolutionary principle for reducing a genome to get the...
You're not being amplified, right?
I'm not being amplified. Okay.
So let's try it here.
And some of these things are really quite structural. In K12, there is an island here, which is almost like a prophage. It's called a pathogenicity island if it were in a pathogen. In 0157, there's a completely different sequence that is integrated in the same place that actually codes for some pathogenic function. So the simplest thing to remove are these big pathogenicity islands. This is kind of a histogram showing the range of size of islands. They go down to this is a log scale. This is 100 bases here. I believe our cutoff was 50 in doing this slide, going up to, I think 80,000 is the biggest. So there's quite a range of size there. And within these regions that are dispensable in the K islands, there's a lot of transposable elements, or recombination hotspot sequences are a kind of probably a former transposable element. A lot of prophages, phage remnants, other cryptic genes, a lot of uncharacterized genes. And amazingly, these are all things that are analogous to or homologous to known virulence determinants in other organisms. They say E. coli is K12 is a pathogen waiting to happen. So getting rid of these gives us a advantage of potential improved safety in drug production, things like that. This IS hopping is a lot bigger deal than I realized, but in the human genome sequence that was published by the U.S. government consortium, there were 18 IS sequences that had hopped into BACs that they had sequenced. And if you there's a paper that the reference is on the next slide, looking at GenBank EMBL database. Kovarik did this work and found 1 in 1,000 of the eukaryotic sequences in that database have some kind of an IS. So 1 in 1,000 of your plasmids will have an IS in it, I guess, if you sequence it. And we took some PCR from plasmid preps, and there, no matter how well you purify the plasmid prep, there's ISs that you can see. So it is really impossible to produce a really correct preparation of a plasmid DNA in regular E. coli. This is the counts that Kovarik got for the data from the database. And so we decided that we would set as our goal, focus on removing horizontally transformed DNA, i.e. the K island, retain core genes coding for normal growth on minimal medium, remove the IS sequences, remove potential virulence genes, adherence genes, toxin genes, cryptic operons, and to do it all without leaving any remnants. Because another problem is you don't want to have a process that will be leaving behind a lot of duplicate sequences or even different sequences at the points where you remove something. This can make the continuation of the process difficult. So this just shows the functions that we chose for elimination. This is a I think this is a plan for the order of removal, so we get the biggest bang for the buck, removing things that are big first. And we got to this deletion of MDS12, multiple deletion strain 12, which we published a couple of years ago, which illustrated the principle and got rid of the 12 biggest islands. And then we spent the next 2 years working this out, finding regions, building primers, developing technology, and we finally got to one that has deleted 40 segments. And these are gene chips run on the DNA to confirm it. Interestingly enough, when we got this finished, there were still 3 IS sequences in it as determined by the chips, which we had to chase down because they had hopped while we were making the thing. And there was also mysteriously a phage that we didn't know about called Phi80 that had hopped into this. And we got a chip that has all the ISs known to man from a database, all the phages, all the drug resistances, all the plasmid origins, and all this stuff. And it's just astonishing when you just look at a strain, even some commercial strains, that they have things in them that nobody has had the ability to look at. So these nimble chips are very nice as a way. It's too bad they're so expensive, but it's sort of a way you should, it's something you should do after every construction. So this is all the stuff that's deleted. The percent here is 14.25, and then we have 41, 42, and 43, which push this up to 15 by just deleting things that we wanted to get rid of, like we wanted them to be T1 resistant, we wanted to get rid of NDA, a few of the standard things that people want to see in a bug. And this MDS72, which we're on our way to, will have deleted 20. You can see that the efficiency of removal, or the quantitative amount, goes down if you choose to do the biggest one first. So now we get to the data. This is the doubling times as a series going from the wild type, number 12, number 31, number 41. And this is the error bars on the doubling time data. So in minimal medium, which is what we kept selecting for, the growth rate just stays the same. In a rich medium, actually it slows down. It's probably losing some of the catabolism things for things in the LB. When we now measure the hopping, and we have a couple of different methods for measuring it, but one that is nice and traditional is that there is a cryptic operon called BGL, which makes E. coli able to grow on salicin. And so one way that you can get salicin, bugs that will grow on salicin, is for one of these ISs to hop into the promoter, and then it activates the gene. And this is the spectrum. Most of the salicin-capable bugs have either IS1 or IS5, and then there's this other set, which are up promoter mutants, point mutants, basically. Now another, oops, I always go backward. Another way to measure mutation rates is to look for cycloserine resistance, which is a transporter that transports the drug in, and so the mutant that makes it resistant tends almost always to be some type of a knockout of CYCA. So again, in the deletion strain, the part of that knockout response that is owing to the insertions is completely eliminated. The little red thing is very small deletions, and then the white or yellow color represents point mutation. I think it's really interesting, and it didn't exactly surprise me that basically the contribution of IS hopping to spontaneous null mutants is about a third of what you get. Then this is a measurement of basically the mutation rate to point mutations of the deletion strain done 7 times. A lot of statistics, and this is sort of what the variation is, which reaches the conclusion that basically there is no statistically significant difference, or a very slight one, in just the mutation rates other than IS hopping in the MDS strains. I forget whether this was 41 or 42, but they're all pretty much the same. Now, when you actually look at gene expression, and I've done a lot of gene expression experiments, and I will just summarize them by saying it's really remarkable that not much happens as a result of the deletion program to change gene expression. There's 3 or 4 things that happen that we think we know the reasons for, but basically it seems like from a system standpoint it is locally homogeneous, so that the regulation works even if you cut pieces out of it, sort of like the brain. And that may be part of the principle of the evolution of these things, that as these genes are moving around, they have to work in their new environment, so the things that are needed to regulate them properly are condensed. But this was a very interesting one, because I looked at the genes that were expressed when you made high levels of protein off of a plasmid, recombinant protein. And I got a little suspicious of this, because I learned early on that heat shock induces the IS transposases. So I wondered if other shocks would do this. And as you can see, that is the case, that the very induced genes include the transposases for IS3, IS5, IS150, integrase of P4, other things that are indicating to me that these stress responses activate the ISs.
This is not heat shock, but something else.
This is overexpression shock. But I was introducing it from the standpoint of heat shock that led me to think stressful conditions would induce these things. And we never did check a lot of different kinds of stresses that we put these bugs through in everyday life, but or in the lab. But what happens, I think it's very beautiful and very logical, is that if there's a lot of protein being expressed, it unleashes sort of a shower of low frequency but very strong knockout. So a cell that has a virus or something that has infected it, if it can by chance hit that gene, it will protect itself from the invader while at the same time not damaging its genome that greatly as it would if it just started mutating everything. So I really think that these elements have a role that is useful in evolution but not useful in bioproduction of protein. And in fact, there are a lot of, if you really talk to fermenter people, they often will find that suddenly the thing they're trying to produce stops and it has an IS in it. At least I've heard tell of such stories. So then we did an experiment to see whether heat shock or cold shock actually changed the mutation rate. And in fact, although it isn't as dramatic as it is the case, that if this is the control, heat shock, you double the amount of cycloserine resistant, I forgot to put what this is, cycloserine resistant cells, and cold shock even more so. And point mutations, that isn't what happens. They go down at shock, or maybe no change. And not much of an effect on these little small deletions. So, for what it's worth, the mutation analysis seems to follow this idea. Now, and I'm getting near the end. I don't know whether I'm running out of time. This gentleman, Dr. Kyle, in the animal vaccine development program in Germany, saw that MDS-12 paper, and he told me this story, that he could not clone this gene for a viral protein 60 because every time he isolated a clone and then sequenced it, it had either IS-1 or IS-5 in it. It was literally so bad that he couldn't get the clone. And so he took MDS-40, and then, of course, there was, I mean, you really have to wonder what was happening if there's no copy of the IS in the genome and it hops in. So he managed to get the clone. Everything was fine. So then I suggested, why don't you take the good clone and put it back into E. coli and see what happens. And by God, he did 6 into DH10B and 6 into C600, so that's 12, and he got 7 patterns. In other words, here is the clone the way it should be, and then these are all the things that individual just randomly picked colonies produced from this IS magnet. And these are the sequencing data that show this is an IS-1, IS-5, TN10 also in there. So that is another practical benefit of removing ISs. This is a transformation experiment. It shows that all 3 of these strains, MDS, the parent MG, and DH10B, have the same magnitude of transformation efficiency with PBR 322. It seems that they're actually a little bit better for large DNA. This is a BAC. And I think one of the things we want to do with this strain is to get it used for BAC production so that we don't have these hops into the genome databases. This is one we don't need to go into. Okay, so now my last 2 slides are attempts to see how well this performs as a organism to increase yield or to use for making large amounts of protein. So this is a fermenter that Dr. Harcum did, in which we were able to get the OD up to 100, and the dry cell weight in grams per liter up to 44, which is considered to be pretty good. And this is one of these fed batch deals where you add the glucose as you go. And the so then we tried growing a similar culture and activated, we had a plasmid with cat chloramphenicol aminotransferase on the lac operon, and these were induced about here with lactose, and then the cat goes up, and it's about the same for the red wild type MG1655 and the 2 replicates of the blue multiple deletion strain. These are the growth curves, and glucose, once it starts being fed, is effectively 0. And something we found out that I don't understand, but that it's something we're probably going to have to figure out and fix, is that there's this acetate reuptake that takes place. This is the level of acetate as a function of the time in the fermentation. And usually when you run out of oxygen, a little bit of acetate is excreted, and then that gets reuptaken. That all happened. And then the 2 deletion strains put out a lot of acetate at the end, whereas the control didn't. So there's something happened in the regulation of acetate overflow. And so far, we think that we always take these things as opportunities because this is somewhat toxic. And so if we're getting recombinant proteins at a normal level under that situation, if we can figure out how to get it back to normal, it would maybe make more protein. So that is our conclusions. These strains are so far behaving as we had hoped, that the core genome contains the stuff that makes E. coli the bug that we love, and all the rest of this junk can be done away with. And I believe on the whole, the size we're going to get down to of perhaps 3,700 genes is by no means small by a lot of standards, nor is it simple, but it does seem like it will work well. So thanks very much for your attention.
Okay, any questions?
Yeah.
It has the same doubling time, Fred, but does it make smaller colonies, or are they the same size?
Colonies are the same.
Okay.
They look pretty normal. In fact, completely normal.
A question over here?
Yeah, sort of along those lines. Hi, Fred.
Hi.
Have you tried to complement this approach with a rational design efforts, selecting, I don't know if this would work, but hands up who else hasn't thought of this, right? Looking for faster growth, for example. If you had an E. coli that doubled every 10 minutes,
Right.
for example, would that be, be fun to find out what that was about?
I'll answer that question in two parts. The first part, is there any attempt to be rational? What we are, one of the things we did rationally was remove the flagella and the chemotaxis. And that frees up energy, and it has an interesting side effect that the cells are easier to spin down. They don't try to swim away. As far as evolution in culture, basically we have not done any culture evolution ourselves, but we have been working with Bernard Paulson on a strain evolution project in which he started growing them on glycerol, which they grow slowly on, and then speeding it up. And we have also done a couple of deletions in MG1655 that make a system that should, as you evolve it, increase lactate production, and that seems to work. And so we have built the same thing in the MDS strain. So one of the things on our agenda is to see whether the in-culture evolution, both on glycerol and on this in this lactate system growing on glucose, well, whether these things evolve more slowly, evolved on a completely different path, whether they will do exactly the same. So the answer is not yet. Something back there?
Yeah. Do these, do the genes that you've deleted, do they tend to have any, can you hear my question or not?
I can hear you
Okay.
fine, I just don't know where you are.
Oh.
Oh, there you are.
So these genes that you've deleted, or these island sequences, do they tend to have any kind of operon bias and operon structure? They tend to be...
Operon bias or
Right.
operon structure. Usually what we have done is to pick a region that has some island or something, and then try to extend out as far as possible until we get to a clear stopping point, and then make it and then see if it works. And so usually operons are always, if we can recognize them, we delete the whole operon. And if there's a gene to catabolize some unusual substrate that we're not that interested in, we'll usually delete that with the rest. But we haven't gone directly to remove catabolism genes. But there isn't anything that I can think of to answer your question about the operons.
The transcripts tend to be, polycystonic.
Can you use the mic, please? Oh, sorry, you don't have it.
Do the transcripts tend to be polycistronic or not? I mean, those genes that you're deleting.
Actually, this polycistronic business is a little over-exaggerated. Most of the genes of E. coli actually are just singletons, but the flagella was a real big operon, and I don't think there was anything particular, systematic or anything. I never really looked at it with an eye toward answering your question, but there's nothing that stood out. Over there?
You are shooting for an organism that is, you said 3,700 genes or so, and that's still a far cry from what they have with mesoplasma. So what are you leaving in that they don't have?
I mentioned those at the very beginning. Things for anaerobic, a whole load of regulators. It's a metabolic generalist. It has 2 or 3 different ways to do recombination. So there's a whole lot of redundancy there, which is no reason to get rid of unless you're just going to have a philosophy about it. If it works, I don't know. Go ahead. And incidentally, I didn't mean to say that I would not go below the 3,900 or 3,700. The 3,700 is what evolution is telling us nature has selected to be a kind of good E. coli. Now we could start to strip it further.
In terms of the folding of foreign proteins in these strains, like
Right.
say higher eukaryotic proteins, do you see any differences at all? And I guess one of the reasons I'm asking is, are the chaperones all maintained in these strains? You're taking some of them out, or what?
We're leaving them in unless we remove them by accident, all of them. And a lot of people add more for these types of strains. So I think they're very important, but we haven't done any experiments to measure them. Function. I think there's probably some chaperones that are good for one kind of protein, some good for another. Maybe there's chaperones that are specifically good for folding, refolding, heat-shocked proteins. Maybe some of them function right at the time it's being synthesized.
Okay, we have a question down here.
I can just shout the question. You mentioned electroporation normal are better than wild type. If it's better, can you explain why?
I certainly can. I have no idea why.
Can you repeat the question, actually?
Oh, the question was, why is the electroporation better, or if it is? And this is a little bit of a variable property. When you do a bunch of electroporation prep, sometimes they work and sometimes they don't. So I don't want to make a big deal of this, but you can certainly make as good a electroporation preps with this bug as you can with anything else, and by tightening up on the being sure it's all done very reproducibly, we're sort of homing in on a very good protocol. But I don't know why electroporation works in the first place.
Could you imply there's some change in physical characteristics?
Huh?
It implies that is there some change in some physical characteristic that's why it's better?
Well, one of the things that obviously helps is the End-A mutation, which is something that knocks out a nuclease. This whole DOR thing that is in the literature about DH10B, there's a flaw in that because the entire 20,000 base pairs of DH10B around DOR is wild type. So there is no DOR mutation. But I sort of chased that for a while to see if that could help. And that's the only thing anybody's really said helps. So it's a mystery completely. The conventional answer is wrong, and there is no other answer.
Okay, why don't we thank our speaker one more time?
Sorry, I was a little
And we'll
flip.
now recess for a short.
So there are a variety of natural systems, natural biological systems that perform some sort of computing function. And of course we know from early work on DNA computing and work on protein interaction, protein computing, and also what I'll focus on for this talk is cell-based computing, since that seems to be a unifying topic in this conference. But certainly the principles of computing in nature go much beyond this. They extend to whole organisms, interorganismal interactions, and to whole communities as well. Different kinds of essentially, and with computing I mean an algorithmic process, that recurrent algorithmic process that has very little essentially outside influence. And so, of course, we've been applying, and we've been engineering biological systems for the production of better biologicals, pharmaceutical proteins, natural products, for example, various kinds of metabolites, for about 25, for a number of years, and engineering these systems for better and better production and better compounds. And so now we're proposing to apply similar technology to engineering for information, computing applications, for materials, new materials applications, and maybe energy applications. And so, it's these non-biological applications that seem to be the focus, essentially, of or at least one focus of synthetic biology, this engineering focus. And so it seems natural to ask, what can we learn from this effort, essentially, this engineering focus over the last 30 years or so, that is relevant for this area?
Just your mic here.
Thank you. And so, well, I'm very pleased to see this direction developing, because the sort of the non-bio nanotechnology direction that has been discussed quite widely for many years now, for biologists is not very satisfying. Essentially, it feels much more, for biologists at least, it seems much more logical, since everything in biology is essentially defined at the level of nanotechnology, is molecularly defined. We feel biology is a very important part of nanotechnology in principle, or example for nanotechnology to follow and to copy. And so we'd much rather build on the 4 billion years of existing design that biology can provide us. And so we'd like to look at existing biological circuits and use these as a starting point. And also look at I think maybe one thing that didn't come up as much as this morning, but I think it's a very important topic of discussion, it's actually not just taking the same old rational design process forward and applying it to biological systems, but I think we should look at the existing evolutionary processes of biology and thinking what these processes of biology can do for engineering, and how that can be applied. Since these processes have led to exquisite complexity, beyond that, essentially what has been possible to be man-made by rational design. Now, there's also some disadvantages that appear to be present with the biological approach, and so I would predict that rational design of a similar complexity in terms of circuit board layout is going to be more difficult with biological systems, because the number of interactions, the number of floating pieces, the number of moving parts that are interacting is so much greater. And so the specificity, by not being spatially limited, the specificity has to be much greater. And that's going to be a tremendous design challenge going forward. And so because of these increased number of interactions, the design challenge increases, and the question is whether this requires a fundamental change in the design process. And so when we look at the sort of the main premise of what people are describing, taking this parts list and building, essentially designing a genetic circuit by rational design in silico first, and then getting parts from this shared parts inventory, which is a wonderful concept, and then building these circuits actually in a cell, testing their performance and fixing the problems essentially one by one. This is, of course, something that biologists have been doing for biological applications for 30 or so years, ever since recombinant DNA has been invented. And so we have a good feeling for what kind of things go wrong when you do this, what works and how often it works and what goes wrong. And so these parts that are described as having well-defined performance, this is, of course, very difficult to do, because it really depends on the context of all of the other thousands of genes in the cell. And so you can never really, in terms of quality control, it's very hard to actually guarantee that these work in the environment, because the environment is so complex and also going to be changing from, so this is very hard to guarantee, much harder than with the traditional hardware. And so, because everything interacts in solution, the specificity quickly becomes limiting. And so you can look at a natural system and you see everything working just fine. But it's hard to appreciate that this took billions of years to come to fruition and to essentially for all the problems to work out, to work themselves out so that everything can interact without causing problems. This is a tremendous design challenge, and there's a tremendous amount of information content involved in avoiding those interactions. And so when you start changing things or putting random proteins into cells, you have to be prepared for large numbers of interactions that were not anticipated. And the question is, do you have time to essentially sort all of these out one by one? Are you going to take single components and sort out each problem one by one? And from experience, we know when you've evolved an enzyme by directed evolution to have a much greater potency, when we try to interpret, we have like 10, 12 mutations. We can't even interpret these single mutations. So even going backwards, so to predict ahead of time, or to analyze essentially what is wrong with your proteins, why it doesn't work, is a tremendous amount of work. And so it's very important to, at the outset, to design the strategy such that you avoid, that you essentially have enough numbers. That's typically how we solve it. That you have enough numbers, essentially, enough different variations are being tried in a systematic fashion to blow through those problems, and that you have a selection for the ones that actually work. So you don't have to go in and try to understand these problems, because these problems are typically not very interesting. They're just unpredicted reactivities. Something binds something else for unpredicted reasons, but it's not interesting to solve. It's not something that because all of these interactions pretty much are, as a class, are known, and it's not something you want to spend your time on. And so, in a sense, this becomes the number of interactions, the number of things that can go wrong at a larger scale, quickly become beyond rational analysis. And so, now in biology, this has been, in terms of biology, natural biology has solved this problem, has found a way to design cells where the pieces all work together and don't kill the cell. And the question is, how does this work, and how can we make this work in the future for complex circuits? And so, I'm sure, biological evolution is effectively the only process that has given rise to to extreme complexity. And so we have not been able to implement this process in other ways yet, like for example, with genetic algorithms. We haven't been able to have the same sort of complexity by mimicking this process yet. And this is, of course, a major goal that we'd like to achieve. But first, I think we have to understand this process better. And I think this topic, this synthetic biology topic and this genetic circuitry topic is a perfect opportunity to understand the process of biological evolution better, to apply it to engineering rather than to biological compounds. And hopefully the evolutionary logic will itself translate to engineering and will be applied to also to non-biological systems. And so if I were to start this, I would start essentially from the billions of complex functioning computers that already exist. Use these as functional starting points. And then evolve these gradually toward desired functions. And so I would use as much as possible existing genomes, but also sets of existing parts that have been shown to work together well, and if necessary, individual parts. But as you go smaller and smaller, the risk increases for trouble. And so it's important to be as conservative as possible, to keep the organism functioning as much as possible without increasing the risk of knocking it out. And so it's important to avoid, in fact, de novo construction as much as possible. If not necessary, there's no reason to take this risk. And so the way I would go about this is to mimic the natural evolutionary process and also the process of breeding by creating large numbers of complex combinations using conservative combinations, so complex combinations of conservative modifications. And this is effectively how in biology these changes are made, and how you go from one generation of functional individuals to another generation of functional individuals that actually have thousands of modifications. But because each of these modifications is relatively conservative in function, you can create complex combinations of them and find a gradual path of improvement. And so I would apply recombination of closely related genomes using natural diversity of organisms, and then recombining either the whole genome or just specific pathways or specific genes. So it's important is to decide at what level do you want to reprogram these organisms, to which extent, and this of course depends on rational information. And so one would typically use different tools to operate at different levels. One would essentially rationally target a certain number of genes, but then and rationally design the libraries for the kinds of changes that you want to make. But in the end, because the interactions are not sufficiently predictable, you'd actually implement them with libraries. So libraries of 10 to the 6 or so different variants of each part, for example. And then after each step, select the parts that work best. So a variety of other approaches that one could use. It's a very conservative approach is to knock genes out rather than knock things in and bring new genes into a cell, which is less conservative because you require this new protein now to work, to be expressed, and to be folded properly. To knock something out is very simple. To knock a single gene out, and you can make very complex combinations of knockouts that are or partial knockouts also that are still quite conservative, and so you can create very complex combinations of conservative effects that together get you to the phenotype very efficiently. And one way of doing this in a targeted fashion, where you only target those genes that you want to manipulate, is using, for example, inhibitory RNA or also antisense RNA. In other cases, you may want to create redundancy of genes so that you can evolve some for new functions, whereas leaving the other ones for housekeeping functions, so that they are not affected. So the typical approach that we have been applying this as a company, also commercially, to commercial projects, mostly for industrial microorganisms, is to take a population, and this is essentially mimicking the how natural evolution works and also how animal and plant breeding works, is to take a population of these existing biological systems that have similar functions, similar sequences, similar functions. So typically closely related species. And the population diversity for each of these species contains up to or around 10 to the 6 variants or so of each of the different parts. And this diversity is the single nucleotide polymorphisms that exist within this species. And so by taking a collection of diversity from diversity of natural organisms and then homologously recombining these, now there's a choice here. At which level do you recombine these? Single gene, pathway, chromosome, whole genome, and then screening for the improvements towards desired function and eliminating the least functional combinations and replicating the best combinations, and repeating this process for many cycles. This process, this evolutionary process applied to microorganisms, is still only samples a very small fraction of all of the potential combinations that could be created. But over time, what's happening here is that the bad parts, the parts that are incompatible with other parts, are being eliminated. And the good combinations of parts are essentially tend to migrate together. For example, will be co-localized on plasmids or on integrons. And so this is a strategy that you see widely happening in nature, where pieces that work together travel together. And so they are essentially they tend to co-migrate and not be linked. They occasionally unlinked by a crossover event, but at a very low rate. They tend to travel together because they work well together. And so the good combinations essentially become closely linked into sets that work well, and that these are sets of parts, just the way you would want to design them. And we see many natural strategies for creating those sets. And so integrons, for example, you see combinations of resistance markers that are combined into a set and that travel together, and also conjugative plasmids similarly. And so importantly, we have been running this as a company now for about 10 years, and there are many valuable applications of this process. And so this technology can, and also in the future, can be further developed without any government funding, which I feel is overall is a very, very positive way to go. And so, okay, good question. But it's a fraction of the total that's... that's a fraction of the total that was spent but so also in the future, I see these kinds of in the future new technologies along these same lines for these different levels of recombination, and also these different commercial applications. There's a clear path forward to essentially build further layers of technology and scope this out much further. And it's essentially economically neutral path can be created, which I think is a very important characteristic for choosing a project. And so couple trends at this, there's a couple opposing trends at this conference, I think, and this is very interesting, actually. We see natural systems having been essentially developed and evolved over 4 billion years. And in contrast, we have systems that were rationally designed by man, complex metabolic systems, and we have a much shorter experience at designing these. And so the question is, what are the building blocks that we're going to use, and what is the design process we're going to use? And so we can use biological building blocks in a rational design process, and that appears to be a strong trend at this conference. But we can also use, and I hope in the future, essentially man-made building blocks and an evolutionary design process. Or we can use an evolutionary components, biological components, and an evolutionary design process for non-biological applications. There's a variety of ways to combine this. And the trend, for example, a small genome might have advantages for a rational approach, but a large genome, I would argue, will have advantages for a evolutionary approach. And so when we look at the process of natural and directed evolution, and we look at this as a process of computation, in that it's an algorithmic process that is recursive and requires no intervention by man, that runs naturally. It's a process of diversity generation and selecting of the fittest. Now, if we compare this to the process by which previous speakers this morning were proposing to generate biological circuits, it's rather different, and so it's interesting to compare these two processes. If you want to build a rather simple circuit in an organism, in E. coli, for example, you just have a few parts. You expect rather few problems. We know that from engineering organisms with lac operators and different promoters, etc., and different express genes. You can put a number of components in without having major problems. But we also know that when you make things more complex and things start to interact not so much with themselves, but they start to interact with other parts of the cell in unknown ways. The mRNAs interact, or all sorts of possible interactions that could never have been predicted. And I would argue in the future are fundamentally not predictable. And so then the approach becomes that this is how we typically operate in biology. If we're engineering these pieces, we make a library of something. So we clone in the whole library. 10 to the 6 is a typical number that we transform, and we screen for the one that works, or select for the one that works. And then we make the next element, next parts as a library of 10 to the 6, put it in, select for the one that works with the previous parts, etc. And so you keep going like this to make little libraries. That's a very safe and prevents a tremendous number of problems by doing it this way. And so I would argue that some of the parts you might want to have in a library version, because then it's easy to select the functional ones. Now, what becomes limiting here is that you have to screen or select each library separately after you introduce it for function and ability to work with the previous one. And you can see that it's impossible to actually put all of these in simultaneously because the numbers become too big. Of course, you can go to smaller numbers and you may still be able to get it to work. But now if you start to make very complex circuits by this approach will stop working because the combinatorials just become too large. And so, so that's a limitation of the de novo synthesis process, that you need to now start combining proven multi-part modules. So you have sets of modules that have been proven to function together in some system, have been tried, and then you combine those pieces together to create larger and larger assemblies. And that's actually similar with DNA synthesis by itself, because you need to check each piece separately for function sometimes before you start combining larger pieces to make an organism. And so it's also these side reactions may not show until very late in the process, because you may be able to check, it's very difficult to ensure that they will work in the final context. And so there's a good reason to argue on theoretical basis that you should have the whole system there, all the components there from the beginning, and then try to make it work better and better. Because that last piece that you add may actually have major problems with the first piece that you added. And so this sequential process has major limitations, theoretical limitations. And so that's why in biology, I think, well, I'm not sure that's why, but at least in biology you see these pieces all being together and gradually evolving in function. And so the question is, well, how does biology deal with this issue of the increasing combinatorials? And so the diversity that's used in nature is not random point diversity. It's not random point mutagenesis. It tends to be homologous recombination of pre-existing diversity. And so existing natural sequences that are recombined where each mutation, so to speak, is functional, is already proven to be functional. And you're just creating combinations of these proven mutations. And so whereas creating 100 mutations by this process gives you cancer, creating 100 new combinations of these mutations has almost no effect. And yes, the origin of these mutations in biology is as random point mutations. But they may have occurred 10,000 years ago, and essentially because they were functional, they have simply stayed in the population, and in fact provided important biological functions in many cases, such as protection against viruses. And so I would like to look at an example of dog breeding. Just look at an extreme example of how biology really works, and why biology is able to develop such very complex systems. And so as an argument for why this type of engineering is very interesting, and we should look at engineering complex biological circuits for engineering applications, maybe by this approach. And so we know that we can breed dogs that are phenotypically quite different. Even at 99.9% homology, which is typical, the dogs that you're breeding will differ by millions of mutations. And so in the process of meiosis, you're creating hundreds of crossovers in random but homologous locations. The puppies that result are typically diverse, but also fully functional. And so you know that you can create a wide variety of phenotypes. You can go anywhere in dog space, so to say, with 5 litters of 5 cycles of 10 puppies. So very little screening, essentially, and gives you very high functionality, and allows you to modify phenotype in rather dramatic ways. Very different behaviors, very complex properties that are very far removed from the genetic basis that you have been able to develop. For example, sheep herding by a collie, we've been able to, you can breed that, you can modulate it, and it's technically easy, and yet you're modifying something that is very far removed from the genetic basis. So I feel this is a very nice example of the kind of engineering that we can do in biological systems. Is the kind of thing we'd like to be able to apply to all sorts of all sorts of different objects. This very complex, molecularly very complex process can be directed by a child, and as molecular biologists, we don't have tools that can do anything near this complexity, and this is where we would like to go. So it's just I just want to point out that the tremendous complexity of the molecular basis of this, permutating hundreds of thousands of mutations, is an achievable goal. It's not overly error-prone. It's a very gentle method to manipulate very complex phenotypes that you could not possibly dream of simulating in silico. And so, if you contrast this with the approach that was most widely used and still is widely used, of random point mutagenesis, where you take a single gene, a 1kb gene, and you mutate now, introduce a single random amino acid point mutation at a time. Over many papers have shown that you get, on average, you get about a ratio of 0.1% of improved mutants. So you have to screen thousands of different versions, and then typically people use the single best clone to parent the next cycle. And so per cycle of doing this, you add one useful mutation. So if you want to adapt your parts for function in the organism by this process, this is a very laborious way to go. You're much better off taking parts from related organisms that are functional in those organisms, recombining those and screening a very small number. And number probably is like orders of magnitude lower than by this process. And so if you compare this error-prone PCR process to breeding, in breeding by natural recombination of natural diversity, you can manipulate a million-fold longer sequence containing a million-fold more mutations, and yet the frequency of improved clones is much higher. And so we feel that the explanation for this is that we're using natural mutations that are proven and proven in their context and conservative, and that we are permutating these using homologous recombination. And so we focused on building this into a process, into a commercial process for optimizing genes and pathways and also whole organisms. And that's now been running for about close to eight or so years. And this is a very practical process that we use on a day-to-day basis to improve genes, pathways, and genomes. And so we can reduce the cycle time dramatically, and we can breed across species barriers. And we can start with whatever number of parents we need. And we can express these in whatever organism we want to apply for the screening. And so this is the process that if you have to optimize parts or build libraries of parts, it's very useful to have multiple family members available. In case some of the family or the family member that you want to work is not working, you simply recombine the parts that are in the library, in the parts list, to generate parts that do work. And whatever the length is, this would be the process that you'd use. And so the way this works, you take the parts, related parts that have related sequences, different operators, different genes, different promoters that are closely related, say 80, 90% or so. And you recombine these by homologous recombination, or also you can also do block-based recombination. And then you screen for the ability of this part to function in the context of the others of the circuit. And so then, if necessary, the best parts can then be taken and you then shuffled again. A pool of the best parts is taken and shuffled again for another cycle until the performance is optimal. So you can use also, even if your part works in your system, you may want to optimize and fine-tune or increase the function of this part in the context of the whole circuit. And that's something, of course, that's not as easily capable with current hardware. That is a biological property that you can, within the system, you can fine-tune this part for optimum compatibility with the rest of the system by this approach. And so it's important the outcome essentially of this experiment mathematically is independent of the length of the DNA. These same mutations with the same conservative, more or less conservative nature can be spread out over a whole genome or over a single gene. Mathematically, that has no impact. The separation has no impact. It's just how many mutations are there and how conservative are they. And so when you make a library of random point mutants from this single part, and so you do error-prone PCR to generate on average 1 to 3 amino acid mutations of this part. If you do higher level mutagenesis, the parts tend to be, if you introduce more mutations than this, the parts tend to be inactive. That has been shown over and over with many systems. So you're sampling only a very small part of sequence space right around your starting sequence. In contrast, if you take 4 parts that are from closely related organisms that are, for example, 80% homologous, and you shuffle these, and you create a sparse library in which the neighboring mutants differ by multiple mutations because of the block exchange between the starting sequences. Now you've created a sparse library, and if you screen this library and then focus on the best mutants, then you can recombine these for further cycles to get more higher density sampling between these. And so you can continue to evolve your part for better and better function in the context of the whole circuit. And so there's a variety of ways that you can make these libraries. So there's rational elements to these libraries that you can dial in, block-based functions essentially. You can do a lot of rational design, include also previously known mutants to stack the odds in your favor. And this is where it becomes essentially a mixture of rational design and a library approach. And so in some cases you may not have natural diversity, and then you can add error-prone PCR. Or you can add partial genes that are inactive. You can add rational mutations that have been published, for example. You can, if a lot of mutations are bunched up together, you can unlink them by directed crossovers using oligonucleotides that are spiked in. If you want to insert elements, for example, different numbers of repeats of an operator or a promoter, you can spike these in at any place that you want using essentially synthetic elements that are just will insert themselves there in the overlap PCR process. And you can also spike in exons or introns. You can do computational linking or unlinking using partial least squares optimization. And a lot of this, of course, is implemented using synthetic genes, so that you can use codon optimization and codon libraries to adapt these elements to the optimum codon usage for the organism that you're going into. And in some cases also, you can essentially start these libraries with different compositions of the parts. So you have 2 different parts that you recombine in one experiment, 3 other parts that you recombine. So you have different combinations of starting points of parts that will give you a different outcome and allow you to go faster towards the desired outcome, towards optimization of the parts in the context of the whole circuit. So the variety of strategies from classical breeding, in fact, that are well known, that you can apply to improving these parts. But we're not only interested in improving parts themselves. A major challenge will be to make the parts work together and to shuffle these, to engineer these genomes at multiple different levels. And so you'll want to do engineering of these circuits, these complex circuits, at the level of individual bases or individual amino acids. You'll want to engineer them at the level of protein exons or introns, or at the level of protein domains. You want to engineer them at the level of single genes or single proteins. You'll want to engineer them at the level of pathways or protein complexes. And you'll want to engineer them at the level of chromosomes or organelles, and at the level of genome, of the whole genome or whole cell. But also you can go beyond that. You can engineer these at the level of the microbial community. And so to optimize the genetic and metabolic exchange that occurs. And I'll say more about that later. But so you really need to have tools, whereas the logic is very similar for these different levels, homologous and non-homologous recombination, mostly the native mechanisms. You really would like to turn on and off each of these levels separately to achieve the outcome that you want. And so we'll have to have tools for this. So we focused on developing separate techniques that allow us to choose a target size where there's specific single genes, specific pathways, or whole genomes. And whether we use as a tool, as a modification tool, whether we use point mutation, or homologous recombination, or non-homologous recombination. So we've been working on creating various combinations of these goals, tools that allow us to address specific technologies. And so we've been talking about increasing genome size, decreasing genome size, and maybe I've mentioned briefly potentially increasing genome size. One possibility that I just want to throw out is the possibility for creating a specialized engineering system. This sort of builds on the idea of the specialized ribosome system that was published by Huey and de Boer. And they published that you can make a specialized ribosome with a specialized mRNA where the ribosome binding site is modified. So now this cell has ribosomes that only express one type of RNA, which is your overexpressed protein. And so you can extend on this. You can actually have a housekeeping genome, for example, Bacillus subtilis, that is completely separated from an engineering genome, for example, E. coli. So you want these pieces to not interact at all, so that you have a housekeeping part that you can turn on and off at will, and you can degrade or denature these host proteins if you want. And you want to have a genome that is optimized for engineering and does not have to perform any housekeeping duties. And so this genome, you want to have a separate ribosome binding site and separate 16S RNA, so that its ribosomes only recognize its mRNAs. And so these would be ribosomes that only function for this part. And now these ribosomes don't have any housekeeping duties. You can feed them all kinds of different tRNAs loaded up with all kinds of amino acids. There are no restrictions anymore on having to do housekeeping duties. And so you can now have a lot more freedom to build whatever you want without interfering with this process. Yes, correct. Yeah. Yes. Okay, I'll skip through this. And so, essentially, for each application, you'll have to look at what sort of at what level you want to engineer. But you want to engineer at the level of single gene, contiguous pathways, non-contiguous pathways, microbial genomes, eukaryotic genomes, or microbial communities. And so there are a number of ways that you can focus on specific distributed pathways. So specific, in language of this conference, specific specific targets that you want to modify for function in the circuit, specific parts that you want to modify. So you want to make a library in several parts to make sure that they work together better than before. And so there's ways that you can target genes homologously to those specific areas using homologous recombination in yeast to make libraries in each position simultaneously. And then, so you create 10 to the 9 or so different combinations, and select for the combination that works best, and transplant this into your organism of choice. The same thing can be done at the inhibitory RNA level. The other, I'll skip through this a little bit. We have been doing a lot of work also at the whole genome level, where we try to shuffle whole genomes without any sequence information. This works beautifully. And so with very little work, we can recombine these without any knowledge of the sequence. And so in this example, it was published in Nature a couple of years ago. We're able to improve a strain that Lilly provided us. And Lilly had over 20 years, they had evolved it to a level that was 6-fold higher in the production of an antibiotic. And in less than a year, with a much smaller number of assays, we're able to obtain a similar or even better result using no sequence information, just by recombining and selecting for just 3 cycles, whereas Lilly had used 20 cycles. I'll skip this one. Okay, last one that I really want to focus on. Well, we've been focusing on learning design rules by simplifying biological systems. And yet in nature you see organisms getting more and more complex. And why are organisms getting more and more complex? Well, partially because this increased complexity provides more ways to compete and survive. So if we want to do engineering with these organisms, what organism do we start with? Do you start with a simple organism or a complex organism as a starting point? And if you're using evolution, I would argue that a complex organism actually is a better starting point. You have more different ways to create the outcome that you want. If you want to create a new metabolic pathway, you're unlikely to create it from a simple organism. You have a much better chance by doing this from a complex organism. And so the most complex starting point that we can find is a microbial community. And so let me skip this. This just shows that having additional tools in E. coli, if you want to make E. coli grow on toluene, getting a natural diversity and plasmids and more natural diversity from a microbial community increases the number of ways that you could achieve success in this with this goal.
Okay, well, I'm going to cut you off so that we can open it up to questions. I apologize.
That's fine.
I'm sorry. Before I open it up to questions, though, can we clap? I'd also like to ask the people in the aisles to please move in. Unfortunately, it's against fire codes to have you guys sitting in the aisles, so if you could please make your way inside. And so are there any questions? Have a question back there?
Yeah, I thought that was very interesting, your combined genome approach, where you had the housekeeping genome and the engineering genome inside the same cell but separated functionally. Have you tried to create something like that with the last concept you mentioned? We have 2 genomes, one a small one rapidly evolvable, say, and then a main one to maintain the cell in the meantime. So basically, let one genome do all of the work, and the other one you can maximize your rate of change and see how far you can go.
We haven't, the reason is that I think you can get much of the same effect by just evolving one genome. And so if certain genes you want to evolve and have one copy for householding as housekeeping function, and the other you want to evolve for some special duty, some new special purpose, it's quite easy. The organism is smart enough to just duplicate this gene essentially and do that itself essentially. You don't have to specifically introduce that rationally. And so this is a solution that the organism can find itself quite easily.
More complex network. You might want to do something just where you have to try to put some pieces together all at once.
Yes, correct. I think that's correct. So I'm essentially this is something that we're unlikely to pursue ourselves because we're focused mostly on applying this for commercial projects. Yeah.
Oh, we had a question here.
So as someone who does rational design, I just want to offer up a defense for the disdain that you expressed for it. So one thing you pointed out was that, you said the child can mate the dogs and create all this diversity, but in a few slides later you said, like to learn about, you had a big cross mark over the, learn about biology from simple systems, and then you went on to talk about, wanting to develop new pathways and whatnot about, getting to something that you want. but I think there are sort of two different philosophies here. Like I think the rational approach is probably much more useful in terms of the means justify the end, whereas I think this sort of the black box approach that you espouse may be more useful for the ends justify the means. So
no, I completely agree, and I think the ideal really is to be able to choose from the tools of from each toolbox as the project requires. So it's just simply to complement, each should complement the other. Ideally.
Right, but I guess I'm taking objection to the statement that you can learn best from studying more complex organisms, and I'm saying that no, I'm saying that I agree with previous speakers in that going for the simpler organism, you might not be able to do as many things, but I think you'll be able to learn more because it is simpler.
I think you would learn different things from different approaches. And so far in engineering, essentially, the ability to learn from evolutionary systems has not been there. And I think now, essentially, that is what I really like about synthetic biology. The fundamental value I see from synthetic biology is that now engineers are really using biology, and they find out the good and the bad, essentially. And I think the good will be that it will discover evolutionary engineering as a very powerful way to manipulate complex systems. And that this will complement the existing, the dominating rational design approach. And I think this complement will be very powerful in the future.
We have another question back there.
So let's say I tried to construct a new car by taking a Mercedes and a BMW, and I make a totally new car. Take the carburetor apart from the Mercedes and the engine from the BMW. I put it all together to make another new car that's fully functional. The reason the new car will work is because there's a certain structure about how a car is built that if it's common enough in both of them, it will also make the new car work.
Correct.
The danger, of course, is that if there's something radically different in something in the nature of the design, then if you're a sensible engineer, you make some things recessive so that they don't screw you up, and some things are dominant. So do you have any insight on the molecular basis or on some design basis on how you'd make something recessive or dominant, or how you keep the structure of something?
This is a very nice example. If one car is metric and the other is non-metric, then you have a problem. And so this is the so organisms have exchanged parts and essentially co-evolved for billions of years. And so they have evolved. There are sophisticated structures, integrons and conjugative plasmids that have worked out essentially that are. So the community relations in these microbial communities, or in genetic exchange and metabolic exchange, are finally evolved, and you can build on that. And so it's very similar, I would think, to a human community, where if you want to make it do something different, you don't start from scratch, but you use what's there, and there's with rather limited changes, you can make it do something different.
Can you use the mic, please, if you still
The question's a very specific question. Do you have any insight on what makes a certain gene recessive or dominant when you do this?
No, but this is easily, from an evolutionary perspective, if you have a clear question, it's a relatively straightforward operation.
Yeah, but I wanted to think about it from rational design. When you do mate to human beings.
Well, what you'd like to have an approach that's specifically focused, where you change genes that are, for example, that modulate methylation, for example, and so you'd like to build these in as library elements, so you can work at these levels specifically, so that not just at genetic changes, but also at epigenetic changes. So epigenetics clearly has a lot to offer. I mean, the same human genome can be expressed as 300 different tissues with completely different properties. And so clearly epigenetics can alone, without any genetic change, can be a very powerful tool for modification.
Okay, we're going to take one last question. Okay, Jerry?
Cross a Mercedes with BMW, you're not going to get a Prius.
If you start with a BMW, what is it?
Okay, one question. Can we make it short, please?
Pim, so you describe being able to go anywhere in dog space in 50 pups, but which is complex phenotypes. For the relatively simple phenotype of increasing the production of one antibiotic, you had to screen 25,000 variants, which is comparable to the number of variants that you have to screen for engineering any specific protein function. What needs to happen in order to get down to the 50, which is actually a screenable number in any assay?
Yeah, okay. Well, since you did some of this work, but so you know the 25,000 was composed of 22,000 by essentially by random mutagenesis, and just a few thousand by recombination. And so if this experiment had been conducted directly with natural diversity, you presumably would have only had to screen a few thousand. And so that would have been a tremendous step forward. It was not possible in this experiment because we only had one strain to start with, but it shows the principle. And so that is, I think, the way forward for combining and making these circuits work. You really would like to build them from existing parts, not just single parts, but essentially families of parts, so that you have a much better chance that you only have to screen 10 or 20 and you have things that work together.
Okay, let's thank our speaker one more time. Okay, and we're now going to be shifting gears a bit and talking more towards the technology end of things. And I think something that a lot of us would like would be push-button DNA synthesis. And if we want to build these minimal organisms, it'd be nice to be able to just synthesize their genomes. So John Mulligan will be talking right now about DNA synthesis, genes today, genomes tomorrow.